Great. Well, thank you so much. It's great to be here. It's been a really exciting day of science, and I think we're getting us near the end, so I'll try to stay energized. Uh, there's more soda back there if you need to go grab one to get us to the end. So first, I just introduced the lymphatic system and sort of my perspective when we study lymphatic system. So most of my career has been spent studying this thing on the right, which we've referred to. It's called a collecting lymphatic vessel. And I think this might be the first video showing how they work in this meeting here. And so I hope you can appreciate the necessity of the fine-tuned structure for this function to actually move fluid. Just as if the heart becomes incredibly enlarged or the valves collapse, you're not going to have functional ejection. When the heart contracts, the same thing would happen in this lymphatic vessel as well. And so there's an intimate relationship between the structure and the function of this to move fluid up a pressure gradient. I think I have, did I touch anything? All right, it's time to move to the next slide. So, all right. So I'm going to go back here. The lymphatics play a key role in fluid balance, protein clearance, immune cell trafficking, and lipid transport. And I think, I hope you get appreciation today that these crucial roles in lymphatic function also make them really challenging to treat, because that's why you see chylous effusions, is because all the lipid from the intestine is being trafficked. You also see very specific immune cell subsets that if they leak out, you have an inability to resolve inflammation. And we saw a beautiful talk earlier about how many of those immune cells are the main source of growth factors that grow lymphatics. So we've heard a lot about the clinical presentation of lymphatic malformations. And their varied presentation depending on where they are, maybe when the mutation occurs in development. And I get to go through these really quickly. So we also know that PIK3CA, of course, has been identified as a very important gene that is responsible, this somatic mutation in lymphatic endothelial cells. And there's really three, what we call these hotspot mutations that make up 75–80%. I think this exact slide was on a previous figure on a previous presentation. And so because of that, we've had great success clinically, or I would say certainly success clinically over what we had previously in treating these patients with sirolimus. And there have been clinical trials to really show its efficacy. And then more recently, alpelisib, which targets the P110 alpha catalytic subunit. And so I'm an engineer. I don't actually see any patients. And it's really hard to get to chose an amazing collaboration, but during the day, like it's 30 minutes to get across Atlanta. So the idea is, what can I do in my lab to assist and work with my collaborators? And so we develop models of disease. And so I want to quickly highlight sort of what's out there in the space when I talk about what I mean by models of lymphatic malformations or vascular malformations. And so we have in vitro models. We have the wonderful talk right before this that would be one such example. You can do things simply like scratch assays or these 3D spheroid assays, and people have used these in patient-derived cells to show responses to therapy in sort of these either 2D or 3D environments. Or you can now, the really first in vivo models were these xenograft models, where you take those patient-derived cells and then you implant them. Of course, to do these models, you have to implant them in a mouse that doesn't have an immune system. And so that presents maybe certain challenges if the immune cells themselves are crucial in the pathogenesis of the disease. But nonetheless, these have been very valuable models to show that these cell types, when implanted in vivo, they have sort of this pathogenesis. And then, of course, we had the really wonderful talk by earlier from Ty Mäkinen's lab, where they've developed a genetic mouse model where they've so far developed it for one specific PIK3CA mutation, where you can induce it in lymphatic endothelial cells. And what they've shown is that in that model, depending on when you do the induction, you get more macrocystic versus microcystic. And then they also showed that spatially, it seems that the pathogenesis is really driven by these cells at the tip. I don't need to repeat it. You heard the talk. It was wonderful. But at the tip, recruit macrophages, immune cells that provide the growth factors that activate the downstream signaling. And of course, the downstream signaling is pathogenic because of the PIK3CA mutation. And so what we've tried to do is sort of say, can we bridge the gap between, say, the simplicity of the 2D model and its limitations and the complexity of the in vivo model, and sort of do something in the middle, and specifically be able to do something with human patient cells where we can better recapitulate the complex, multicellular and mechanical microenvironment to grow patient-specific lymphatic malformations and then look at patient-specific responses to therapy. And so I had at the exact same time, so Josh was a former PhD student, and he had been developing these hydrogels that are synthetic hydrogels based on a pegmaliamide polymer system. He had been developing them to support lymphatic regeneration. And what he had did, I'm not going to go through his data, but it's in this paper, but there's things we can tune with these hydrogels. So they're pretty simple. It's actually mostly PEG and water. But then it's a 4-arm polymer that we can cross-link. And what we can do is we can put these cross-linkers that can be enzymatically cleaved when cells secrete MMPs. We also put peptides on there that allow for adhesion of extracellular matrix molecules that are secreted by the cells themselves. So the idea is you provide sort of the baseline scaffold, and then the cells themselves lay down matrix and degrade the polymer so that you can really get the matrix that would be formed by those cells themselves. This is sort of the idea. So what he did is he took tiny rings of collecting lymphatic vessels from rat lungs and then embedded in these hydrogels, and then we have all these knobs we can turn. So we can change the mechanical stiffness. We can change what type of peptide is present. We can change the concentration of that peptide. You can change how fast it degrades, et cetera, et cetera. And he got sort of our perfect hydrogel. And what we did is this is just compared next to a collagen gel, which is sort of a common 3D gel. And maybe if I could start on the bottom 2 left figures and see, this is a co-localization map that shows the co-localization of lymphatic endothelial cells with lymphatic muscle cells. And so when that is really bright, that's suggesting coordinated migration of these 2 cells together. And so in this hydrogel, you really do get sprouts that have both lymphatic endothelial cells and lymphatic muscle in the hydrogel, versus when you put them in the collagen, you really just sort of get rapid kind of unorganized proliferation out of the core itself. And then shown here is sort of a stain together of this network. And this dashed line here, that's the original embedded ring from the lymphatic vessel. And he went on to show that we could actually put this in a host mouse, and it would engraft into host vasculature. But while this all happened, I got an email from Matt Hawkins. Is Matt still here? I pointed over there. He's not there. Maybe he's left. I got an email from Matt. He's seen this talk so many times from Matt Hawkins. And he introduced himself and said, "Hey, have you ever heard of vascular anomalies and lymphatic malformations?" I was like, no, not really. And so this is sort of what prompted the collaboration is he was like, "Well, I can get you tissue." And I thought, well, let's see what happens if we take that tissue and put it in this sort of engineered hydrogel. And so we did that. And the first thing we did, we got 5 different patients of tissue. And we did our best when we got the biopsy to sort of take the biopsy and kind of cut it down into different pieces that were all roughly the same mass, and then embed those in the hydrogels. And then the first thing we would do is we could evaluate their metabolic assay using just Nalamore Blue assay, or metabolic activity using Nalamore Blue assay. And the first thing we found is we could get pretty reproducible metabolic activity within a single patient, but across one patient to another, there was differences. I can't say, you all know when you do these biopsies, you're just sort of going in and taking tissue. So is this just, well, where did we hit the biopsy? Is it a difference? Some of these I'll show are macrocystic, some are microcystic. They've had different history of whether or not they've had sirolimus treatment. So we don't know like why, but it was interesting nonetheless. And then when we looked, when we looked at the stain, what we found was really it's the lymphatic endothelial cells. So on the right, that's a potoplanin stain that branched out. So these things branch out quite nicely. They're actually it's a great model to teach someone to culture cells on. Primary healthy lymphatics are a pain in the rear to learn how to culture, but these are great. They grow really well. And so we saw that, and then we did a quick study where we looked at rapamycin, and again, this is a small, just 5 patients, but and it could be just the nature of the biopsy, but we found that the macrocystic were very responsive to rapamycin, where the microcystic were not. In this small subset. The disadvantage to this is you get a biopsy, you know, you cut it up, and then you get your experiment that you get to do on that patient, and you're done because you can't go back and keep biopsying these patients. And so, so we thought, okay, but we liked the idea of putting the whole tissue in, because then we're taking the entire microenvironment and all the cells present. It's not just lymphatic endothelial cells. And so I said, well, let's put it in and let's grow it for a long time and see what happens. And so I think this is a patient that's been grown for 30 days. And we got really substantial sprouting. And then what we did, so then we could actually take these, and we've done this for 3 patients. I only have the results from 2. We could send this for whole exome sequencing. So, I mean, if anything, maybe we have a way to enrich cells. If you have trouble with mosaicism, put them in the hydrogel, then you have, if it has a functional mutation, you have a ton of them 30 days later, because those are the ones that are proliferating. I'll say a lot of these are mutations of variants of unknown clinical significance, but they are pathogenic in the sense of they're very hyperproliferative in these gels compared to normal ones. And so then what we did is we did single cell RNA sequencing, and we had saved half of the fresh biopsy to do single cell RNA-seq. And then, so that's on the left, and on the right is from cells that have been expanded for 30 days. And do I have a, what you'll see here is actually, it turns out this is an expanded map of that, but what you'll see here is this population here is sort of what explodes 30 days later. You do still have in the hydrogel other cells present 30 days later, but really where you have expansion are lymphatic endothelial cells. And when we did this, we did the bioinformatics, we basically matched cell numbers between the fresh biopsy and the expanded one, which means we had to downsample the expanded one because you get so many more cells out of that to do the clustering. But then what we could do is look specifically at the clustering of the expanded cells, and we see these different lymphatic endothelial cell subtypes still persisting in these. And so what this gave us sort of some confidence that, well, maybe we can take these expanded cells and use those as a source for a bank of lymphatic malformation endothelial cells, but lymphatic malformation endothelial cells that have never touched 1 gigapal stiff tissue culture plastic. Because we know, like we talked about mechanical transduction, and we all know the moment you throw your cells on it's an infinitely stiff piece of plastic, the cells are going to differentiate. And so that's Josh graduated, he was ready to move on. And so Jarlis, I was lucky to have her come into my lab, and that's where she took off, or she came in. And so she ended up developing a way that we could take these, expand them. So these are 3 different patients there. So we grow them for 30 days. We digest. We actually do selection on podoplanin, and we do we have our lymphatic endothelial cells, and we have other. And so anything that's podoplanin negative goes into one, podoplanin positive into another. And then what we can do is reassemble these into spheroids, embed those spheroids into the hydrogel, and then these are structures that we get. I think this is probably 10 days after growth. And the nice thing now is we've done this from banks of cell lines that are patient specific, where we can now grow and do reproducible experiments where we do drug screening. And this is in this bottom one, the purple here is actually FITC, and so we FITC dextran. And so we can inject large molecular weight dextran molecules, and they perfuse into the sort of, I don't want to call them lumen, but we'll say open spaces within these hydrogel branches. And so the first thing, I mean, maybe it's because I'm a mechanical engineer and it's my bias, or maybe because it's important, we thought, let's think about stiffness and how that might affect the phenotype of these cells. And we were inspired by another paper from Taya Mack, and I guess I'm a fan, that was Make a Fry did this when she was a PhD student in her lab, but she showed that stiffness seemed to be a cue, or proposed that stiffness was a cue in the early migration event of lymphatic endothelial cells from the cardinal vein off as it budded out and differentiated into lymphatics, and that it's much softer outside the cardinal vein than inside. And so we thought, well, let's look at how stiffness affects. And so what you'll see here, these are the 3 different patients that we've that we've done, and we do them at different stiffnesses. So 6% is the softest, 12% is the largest. Although I will say that, I mean, the stiffness range here is still these are very, very soft hydrogels. What we found, we can quantify that. The first thing you probably saw quite strikingly is that one malformation patients grow much faster than another and much sort of branch much larger than the other. If we look specifically, here's just shown quantifying the branching length. And then if we look at stiffness, for two of the patients, they really didn't see within this window of stiffness range much of an effect, but we did see an effect of stiffness on the LMO3 patients. And so then what we did from there is we decided to do a drug screen. And so the way this assay works is you grow the sprout first. So the top left, this is a spheroid that has fully sprouted. And then we treat it with a specific inhibitor for 24 hours. And then we can capture the regression of established sprouts. And so these are the different inhibitors that we've talked about a lot today. And so and then the way this data is shown is it's normalized relative to untreated. So 0 would be no regression, and that's for that particular patient untreated. And so the larger the bar is, the larger the regression you get. And we really had 3 classes of inhibitors. So we had 2 different inhibitors of phosphorylation of the growth factor receptor. One is specific to VEGF-C, or VEGFR3, sorry, and one is blocks VEGFR1, 2, and 3. And then, of course, we did sirolimus, alpelisib, and then capivasertib. And what's interesting is we see statistical differences in sort of between patients. And then this is just one example of, like, with each patient, which inhibitor was the most effective at really reducing sprouting within, again, a 24-hour period of treatment. We also within this can look at cell death. And so this sprouting is the live cells and just counting the complexity of that network. We have a dead stain. We can look at cell death. Interestingly, we don't have it, and a lot of this because the way the hydrogel is, when the branches regress, those cells are gone. So this is remaining cells that are dead. You basically, the core is still present and those cells are still alive. And so you don't see a lot of cell death in patient 3. You do see some cell death in patient 5 and 6, and the VEGF receptor inhibitors seem to be the most effective. And then the last thing we did is because we have this spatial information, we can look at where the death is occurring. And what we found was, in the cases where there really were differential and strong effects of the drug, it was really because the effect occurred at the peripheral region and of the sprout as opposed to the core. And so one of the things we're interested in here now is, we can remove these inhibitors, and if you have this core that did not die, that seemed to be in a sense resistant, of course this was only after 24 hours, can you get sprouts to be reestablished? And then can you not only define efficacy as regression, but efficacy as persistence of the lack of formation of new sprouts? So that's kind of where we're at. I'd love to think specifically, I think, because people are in this room that probably decided to take a risk on an engineer and give us some money before we had done anything. And so the Orphan Disease Center and $1 Million Bike Ride helped us get this work going. And then my collaborators, Matt Hawkins and Mike Briones, who are here with us, as well as other collaborators in this work. Really, Jarlis is a very talented PhD student who will be graduating and looking for a postdoc within this year. And so that's a plug for her. And Josh was a former PhD student. And so thank you very much. Happy to take questions.